Now, what I want to talk about is the epidemiology of Parkinson's in New Zealand. This is research that we've been doing over the past 8 years, and two other people involved in this research. Let's just get this focused. There we go. There's Tony Pitcher here. She conceived this epidemiology research program, and also Tim Anderson, who is the movement disorders specialist neurologist involved in this project as well. So the background and aims. What we want to achieve with this is to estimate the number of people in New Zealand with Parkinson's and how this has been changing over time. Also understand the influences of sex, age, ethnicity, and region. And following that, understand risk protective factors and what it can tell us about the disease process itself. So how do we estimate the number of people with Parkinson's in New Zealand? Well, we primarily do that by looking at the prescriptions that people get. So every time that someone gets a prescription, the pharmacist enters that into a database, and then we can get those records from the Ministry of Health. Now, people with Parkinson's take a certain range of medications, so we can identify those medications. And those medications aren't just used for Parkinson's disease, so there are other conditions that they are used for. So what we do is we use a range of other data sources to see, okay, does this person have Parkinson's disease or do they have something else? And this includes NZBRI, which is used for home care, long-term care assessments, the National Minimal Data Collection, which is hospital admissions, where they get diagnoses attached to that admission. Mortality, so on the death certificate, you have a whole bunch of diagnoses attached to it, and also outpatient and health databases. We then do some mathematical modeling. So I'll try and describe this using some pictures. So here, what we look at is that given your age, ethnicity, and sex, and the medications you are taking, what is the probability that you have Parkinson's disease? So in this case here, we're looking at a particular medication, levodopa. And if you're 20 years old and taking levodopa, you can actually be taking it for some other conditions, and you only actually have a 50% probability of having Parkinson's. But if you're 60 or 80 and taking levodopa, it's almost 100% probability that you have Parkinson's disease. In this other case here, this class of medications, if you're taking it at a young age, it's very unlikely that you have Parkinson's. While if you're taking it when you're 80, then you've got about a 50% chance of having Parkinson's. And these colored people here represent the probability. So the darker color means a greater chance of having Parkinson's. And over here, these all represent a low probability of having Parkinson's. So the output from this is we give everyone an individual probability of having Parkinson's. We can then sum all these individual probabilities together, which gives us a total number of people with Parkinson's. You can stratify this by age, sex, and ethnicity. We can then correct for missing cases, and then we can also standardize by the number of people in the country, etc. So what does this tell us? So here is a look from 2006 to 2019 of the new cases of Parkinson's. Overall, countrywide, in 2006, there was about 1,000 new cases in that year. Going to 2019, it's increased to about 1,250. It fluctuates a little bit over the years, but there's been a general trend of increasing. More cases in males and slightly flat line of cases in females. We can then look at the number of people with Parkinson's. And again, that's been steadily increasing from about 7,000 in 2006 up to about 11,000 in 2020. And again, greater number of males and a slightly less number of females. So this figure here is a balance of the number of new cases and life expectancy as well. We can look to see the influence of age, and there's a very sharp rise from about age 50 up to about age 80. And then after about age 80, we actually see a drop off in numbers. There are a couple of potential reasons for this. It may be that the pool of people who can potentially get Parkinson's disease gets depleted. It could also be that past that age, with other health conditions going on, etc., these people aren't diagnosed with Parkinson's as frequently. We're uncertain about that, and we're actually looking into that further. Again, looking at prevalence by age, so this is proportion of the population that have Parkinson's in each age group. A steady increase from about 50 up to about 80 and then plateauing a little bit there. This lags just a little bit more than your incidence, and again, a drop off going over to about 100. So what causes this increase over time? Is it purely our aging population which is causing it, or is there something else going on? To look at that, we can look at standardized incidence and prevalence. What we do there is we look at the age-specific rates that we see, and we look at a reference population. So for example, our 2013 population, we set that and multiply each of these age-specific rates for each of the years. And that gives us our age-specific incidence and prevalence. And when we do that, we see that incidence overall has been relatively steady up to about 2014, but then in more recent years has dropped off a little bit. Now that is saying that given our aging population, we'd actually expect slightly more Parkinson's cases. We're not completely sure at this case if that's just an artifact of the data and if we need to take more things into account, or if there's actually something going on and something has reduced the risk of Parkinson's like an environmental factor. Further research is required to see what actually is causing that change in the last few years. If it does turn out to be actually a real effect going on there, that is potentially good news. The same for prevalence. It's been slowly increasing up to about 2014. That shows that the increase has been entirely due to the change in the age structure of the population, with a slight increase in life expectancy as well. But then again, in the last few years, it hasn't followed that increase. Further research is required to see if the prevalence is actually dropping off a little bit, or if it's just something going on with the data. We can also stratify by ethnicity. What this shows is that European is the highest rate, and it's about half the rate in Maori. There are a few potential causes for this. One could be that ethnicity is miscoded in some of the data sources. Another reason could be that Maori don't access treatment or are treated later on. The third reason could be that Maori actually do have a lower rate of Parkinson's. And that is something that we are also investigating at the moment. We can also break numbers down by region. So this is data for 2013. Still some issues with the latest 2018 census that we're working on fixing. These numbers today were about 30% larger. So Canterbury actually has the most people with Parkinson's in New Zealand. That's followed by Waitemata in Auckland, and then the southern DHB is the fourth most. And then it goes all the way down to the west coast, which had approximately 81 in 2013, the lowest. We can then again standardize these figures to the size of the population and the structure of the population. And this is what we get. Auckland, it has the highest rate. And then it goes South Canterbury, Canterbury, Southern, a couple of other North Island DHBs, and then West Coast and Nelson Marlborough. You notice that quite a few of the South Island DHBs, all coded in blue, are actually at the higher rate. One potential reason for that is living in a rural area actually is a slightly higher risk of Parkinson's. Yeah. Looking at the current and predicted number of people with Parkinson's, this is what we did based upon 2013 data. And it is tracking relatively along these lines here. So we have got up to around the 11,000 now, and we are heading towards 20 or so thousand around 2040, 2041, 2042, etc. After that, it's expected to slow down a little bit. This is based upon Stats New Zealand's population structure projections in New Zealand. Right, that's the end of my part of the talk. I think, do we want to take questions for this part now? Yes?
Yes, this cover the blood type.
No, it hasn't. That's not something that we've looked at yet. Not sure if the blood group is really recorded in any of these databases, so it might be hard to actually investigate. There's potential pesticides, etc., which are linked to Parkinson's. Drinking well water is another thing that has been linked. Yeah, I live in a rural section as well, and I have just installed a reverse osmosis filter for other reasons, but yeah. These things don't greatly increase, but they just slightly increase your relative risk of getting Parkinson's.
My rural area has higher risk of Parkinson, and is that triggered by?
Potentially, there have been some pesticides which have been linked to Parkinson's. So farmers in particular, as mentioned before, well water has been linked in some cases to higher rates of Parkinson's.
So agriculture,
agriculture itself, yes.
Analyze the severity of individual cases. Like the sample population are all quite minor cases of Parkinson's.
In this analysis here, we don't look at severity. We do have another study, the Christchurch Parkinson's longitudinal study, where we have a much smaller sample, where we do much more detailed analysis of people. And in that study there, we do fully look at the severity of the disease. Okay, I'll now welcome up Michael MacAskill, We'll now take a wider view of Parkinson's disease.
Kia ora. Just want to acknowledge the work that Daniel and Tony in particular have done over many years. Put that in perspective, that's one of the largest epidemiology studies in the world, actually. So we're in a small country, but that study involves pretty much every person in the country, or certainly every person with Parkinson's. So a lot of comparable studies overseas would involve a few hundred patients when they look at the age structure and the risk factors and so on. And in this study, as you see, it's currently involving 11,000 people in New Zealand, and it gives us incredibly high quality data compared to a lot of other studies. So I just want to acknowledge again the MS and PD Society. We're very keen to support what they do, and I guess there's two different aspects to the way our two organisations work. MS and PD Society very much focused on improving care for people with Parkinson's and MS in the community. And on the research side, I like to say that we are focused not so much on the care side of things, but more on giving people hope, really, which is about the future. Are there going to be better ways of treating or even preventing these disorders in the future? Not necessarily for us, but for people who come after us. And I just also want to acknowledge people at the other end of the lifespan. We've been talking about the diseases of aging. We've got a group of young people here today. The good news for you is that your brains are still growing. Bad news, I gave a lecture this morning to a bunch of nursing students, and so I'm prepared on this. Your brain stops growing about the age of 19, so you haven't got that much longer to go. After that, it's all downhill. We all lose brain cells every day, basically, from about the age of 19 or 20. About 85,000 neurons on average per day. While you're sitting there, that's 1 a second. So as Daniel and I are trying to push information into that brain is getting smaller and less capable of absorbing it. So I'll try and not keep you too long. But today I want to talk about a particular paper here, and I've got a bunch of copies of it, so don't be too worried about taking notes or anything. If you want to, I'll pass these around, and I'll print it out on A3 paper, so hopefully it's reasonably readable. And it's from this fellow here, Professor Roger Brach, who's a very noted neurologist who studies Parkinson's and other movement disorders at Cambridge University. And he just sort of stood back and took an overview, just in 2 pages, really, in an article, and I thought it was useful to share that. This is beyond just the research that we're doing with NZBRI, but to look at Parkinson's in general and how things have really changed over the last 20 years. And at the very end, I will just have one little pointer for some of the research that we are going to be doing at NZBRI. So what I'm going to cover today are the things that he highlights that have changed over the last 20 years. I'll just pop them all up there. Are these. So the first one, well, actually we'll step through them, but it's a variety of ways in which our understanding of the disease has really fundamentally changed over the last 20 years. So Parkinson's has got a history of a bit over 200 years, first described in 1817. And so we're over 200 years into trying to understand it, but really there's been a few revolutions over the last couple of decades. And I think that means that we are going to see some changes certainly in treatment and potentially even prevention in the coming, certainly in the coming decade or so. So let's start with the first one. This is sounds like technical language here, but it's the alpha-synuclein prion hypothesis. It sounds like sort of a science fiction sort of movie. It's almost as exciting. The story starts actually back in the 1960s in Papua New Guinea, and there was a disease there called Kuru, which is a little bit similar to Creutzfeldt-Jakob disease or mad cow disease. So very serious disease that affected people in these very rural villages with a very severe neurological disorder that was fatal. And it took a while for people to figure out how it was spread. And when that problem was solved, it did lead to a Nobel Prize. It turns out that this disease was spread in a completely different way. That we previously, we've always thought that infectious diseases spread through bacteria and viruses, just like we were taught at school. But there was a third way that infectious diseases spread, and that's through something that's not actually alive, unlike a bacterium or sort of a virus. It's through something called a prion. And a prion is just it's just a protein. It's not living at all, it's just a chemical. And the way in which this disorder was spread was from a single case in about the year 1900, it's thought, and it spread through cannibalism. So this was a tribe that still practiced cannibalism until about the 1960s. And so this was a brain disease, and the brains were left to the children and the women of the tribe. The men wouldn't deign to eat the brains, so that was left over for the kids. And sometimes decades later, the effects of eating that brain tissue would result in the disease developing, but also it would actually strike children. And these children, unfortunately, that you're seeing there, passed away within about a year of this photo being taken. So fortunately, that tradition of eating brains stopped at around the year 1960, but the last case of this disease happened around 2005. So 45 years later, this disease was still being seen. But what it showed was that we can get brain diseases that spread, like I say, not through viruses or through bacteria, but through prions, through a certain sort of protein. And to illustrate what we mean by that, this is a good protein that we're all relatively familiar with. Is an egg. Eggs come in different forms. There's a nice runny egg like that. You pop it in a pan for about 30 seconds and, or a little bit longer, you'll get this. Completely changes from being nice and runny to being quite rubbery, depending how skilled you are at frying an egg. The same thing happens with proteins in our brain. So this is a picture of a neuron. We can see this is the body of a neuron and a brain cell, and this is the way it communicates to other brain cells. It's got this big long axon here. But if we just take a slice through the body of the neuron here, we can get a picture like this. This is taken through a microscope. And there's a couple of things to notice here. This is a cell from a person with Parkinson's in a part of the brain called the substantia nigra, which leads to the movement symptoms in Parkinson's. And there's two things to note there. One is that that cell is stained quite a dark color, and that's how we can see these cells under a microscope. The other thing is this little purple blob. And that is a protein, a blob of protein. It's called the lewy body, and it's a clump really of insoluble protein. And since we've known this since about the turn of the 20th century, that this is the hallmark of what Parkinson's disease is the detection of those lewy bodies in the brain. Now, we can't detect those until people have passed away, and we look at them in an autopsy in a microscope. So that's not particularly useful for people who have Parkinson's at this stage. But still, after 100 years, we don't know if those lewy bodies are causing the problem. We still don't know if those are killing the neurons or if that's actually the neuron protecting itself, taking that clump of protein and surrounding it, putting it into a tiny little packet to protect itself from it. But the key thing there is that alpha-synuclein, which is this protein here, and don't worry, we all have it. It's in every one of our neurons, and it's a useful thing. Normally has this sort of structure. It's a big, long, windy thing, and it's effectively soluble, just like a runny raw egg. But every now and then, this is the alpha-synuclein, like I say, you can sort of see it stretched out quite long and thin. Sometimes it'll join together with friends and form a sort of a mat, and it will just sort of go backwards and forwards like that. So it can form clumps, but then break up apart and hang out by itself. But every now and then, this will happen. It bends into a misfolded kind of shape. And now when it finds a friend and it joins together, it will make that friend also bend into that funny shape, and they'll stick together and then they won't break apart. So this lump of alpha-synuclein has become insoluble. And as we saw there, that's forming the lewy body. It gets forms a clump inside the neuron. So that's something that we've known really for about, well, we've known about lewy bodies for about 100 years. We've known what the chemical is for about 50 years. But the real new piece of knowledge happened just in 2008. And around that time, or the decade before that, there'd been experiments with stem cell treatment. So that's putting in cells that come from fetuses into the brains of people with Parkinson's that were designed to produce extra dopamine. So dopamine is the chemical that is missing in Parkinson's when certain cells die. And so it's thought, well, if we just replace those cells, everything would be hunky-dory. Those experiments didn't work terribly well, unfortunately. But they did reveal a secret later on when some of those people eventually passed away and their brains were examined. And what was seen is that these clumps, the lewy bodies, had spread into those new cells. And those cells had come from fetuses, so none of them were more than 10 years old. And so what that showed is that Parkinson's doesn't need to have old brain cells. Brand new brain cells can get the Parkinson's pathology. So it led to this understanding really that this protein moves from one neuron to another. So an old neuron in Parkinson's will quite happily affect a brand new one. So you don't need to be an old neuron sort of on your last leg. A brand new neuron can be infected. But what that showed was that it is really a prion disease. It's a spread of proteins within the brain. So it's like an infection, but it's happening inside the body, and there's no virus or bacteria involved. And it does spread then through the brain. So what is important about the prion hypothesis as being part of the cause of Parkinson's? Well, firstly, we now know what to potentially target. And so there's a lot of work going on in other centres around the world to try and target that abnormally clumped form of alpha-synuclein, that protein, which may slow down or even stop the disease process. And so there's a lot of work going on there. That's being tried in other diseases that have similar processes. Unfortunately, none of those attempts have really borne fruit as yet, but I think people are still trying to solve the problem. But what that also tells us is that there must be a stage of Parkinson's before the symptoms start. And we know that because when a person with Parkinson's first presents with symptoms to either a GP or to a neurologist, that the clump of cells that produces dopamine, about half of those cells have already died before the motor symptoms start. So there must be a period before the symptoms start in which the disease is already ongoing. But the good news there is that means, again, that there could be a period before the symptoms arise where treatment could be applied. So that brings us to Roger Barker's second point here, what he calls the concept of prodromal Parkinson's disease. And prodromal is just a fancy word, but it really means just before the disease starts, which is a strange thing to talk about a disease before a disease. But what it really means is the aspect of Parkinson's before you have any motor symptoms. So before there's a tremor, or before there's any stiffness of the muscles or slowness of movement, that the disease is ongoing, and it may have been so for 20 years or more. So this is the part of the brain called the substantia nigra, which is like, sort of tend to tell people it's just a fancy Latin word for black stuff. And actually, this is exactly what that part of the brain looks like. It hasn't been stained or treated in any way. If you just take a slice through the midbrain, fortunately, this person had already passed away before that happened. We don't do those sorts of studies. And you can see this is a healthy person, actually, and so it's got a nice dark staining. There's only about 100,000 cells or so. This is very small. It's only a few millimeters across there. But that's a nice healthy brain there with good dark staining. It's dark because the cells that make dopamine in our brain have a chemical in them called neuromelanin, which is basically the same chemical that we have that makes our skin brown. All right. And this is a person with Parkinson's, and you can see those dark cells have died off, and so we don't see that typical staining. And as I said, that's at the stage at which symptoms arise, there's already been a huge amount of damage there. So even if we had a cure today for Parkinson's, it wouldn't be able to undo a lot of that damage because it's already happened by the time that symptoms arise. But the good news is that we should be able to identify earlier cases. And that takes us to something which also, again, sounds like the title of a science fiction movie, the Brach hypothesis. And this is named after a fellow called Professor Brach, who's from the Netherlands. I don't know if that's actually how to pronounce his name, because the Dutch have curious ways of pronouncing things. But he did a really interesting study where he looked at the brains of hundreds of people coming through a hospital and taking everybody to autopsy. Now, that almost never happens in New Zealand. It's very expensive to get autopsies here, and we just don't tend to do it. But he took pretty much everyone who died in the hospital of a certain age and looked at their brain. So it wasn't just people with Parkinson's or Alzheimer's. He was interested in those diseases, but he wanted to look basically at every brain that he could see. And what he found was that actually he could see those Lewy bodies in people who didn't have Parkinson's yet. So this is going back about 20 years or so. What he saw was that the disease is actually starting at the brainstem here. This is the substantia nigra up here, but he saw these early changes right at the top of the spinal cord. And so he devised what's called the Brach staging system, where there's actually 6 stages to Parkinson's disease. And where the symptoms arise is actually stage 4. So that's the point at which there's been enough damage occurring in the brain, and it sort of moves up the brainstem until it gets to the substantia nigra. At that stage, we see the symptoms, but we're 4 steps already into the disease at that stage. So as I say, that's a problem that if we come up with a proper treatment for Parkinson's, unfortunately, a lot of damage is already done by the time people are diagnosed. So we really need to find these people here. These are the people before motor symptoms arise. Can we identify them? And that is increasingly becoming possible. It sounds like an impossible task, but can we find people with Parkinson's before they get the symptoms of Parkinson's? And to do that, we have to find those symptoms that arise before the movements start to change. And that takes us to the importance of this thing called REM, sleep behavior disorder. Now, there may be people in the room for whom this is an issue, but effectively, for most of us, when we are dreaming or in REM sleep, our body is paralyzed, so our muscles don't move. But for a few people who have REM sleep behavior disorder, that paralysis doesn't kick in. And so during your dreams, you tend to act them out. So there'll be a lot of potentially kicking and punching and flailing about. And this may sound familiar to potentially some people, potentially more the partners of people with Parkinson's, because you're the ones who are more likely to get woken up by this. And it does seem that the content of dreams for people with Parkinson's, or particularly for men with Parkinson's, is often quite violent. It's about running away from monsters or having fights with people and that sort of thing. It's a common sort of theme, unfortunately. So that particular symptom is something that comes on for many people before the movement symptoms of Parkinson's. And so increasingly across the world now, groups are looking at identifying people with that disturbance of sleep. So this is before people are diagnosed with Parkinson's, people are gathering groups of those people together and seeing how they change over time. And so this, hopefully you can sort of see that at the back, but I'll sort of talk your way through it, but I'm not going to show too many graphs today, but this is one of the more important ones. This is from a group in Canada, and they've done a really impressive study here where they've been following for quite some time now, about 10 years, a group of people with that sleep behavior disorder. So again, these are people without Parkinson's. They're people just who are acting out during their dreams. And so they're recruited on that basis, and then they're followed up every year. And they look at a variety of symptoms that will be familiar to some of you. Things like, what's your sense of smell like? Do you have problems with constipation? What's your colour vision like? And do you have any movement impairments? And what they found was that actually, this is the point zero. These people are increasingly now being diagnosed with Parkinson's. So that's what we call time zero. That's when they present to diagnosis because they've got motor symptoms. And what we see, let's look at the purple line here. This is the loss of sense of smell. And so for people here who are diagnosed, what we're finding is that their loss of sense of smell is very high. It's about 60 or 70% on a severity scale. And if we go backwards over time, we see that that change in loss of sense of smell is going back about 20 years. So the first changes in Parkinson's are happening over that kind of time period. And the next one here that seems to be the earliest one is actually changes in colour vision, which again is not something that we traditionally pay too much attention to in Parkinson's, but I think is quite important. It's something I think that I want us to look at more locally in Christchurch. We then look at another one that will be of importance to some people, which is constipation. Again, not often talked about with Parkinson's, but definitely can be a problem. And that also, again, seems to start to change about 10 years before the onsets of tremor or any of the other motor symptoms that we think about. Now, I won't talk actually about the movement or cognitive signs until we get to the next graph. So this is the same graph actually, but those people who have REM sleep behavior disorder, some of them will get Parkinson's disease, and some of them will get dementia with Lewy bodies, which is a very closely related disorder. But this is one where you'll get the symptoms of Parkinson's, but more early on, there's likely to be cognitive changes. And we see a little bit of a different picture here. So when we look at just the people who get Parkinson's, there's this rapid rise in motor symptoms. That's not too surprising, about 2 years before diagnosis. So that's when people will start to notice that maybe they're getting slower, that walking is slowing down, and maybe there's the initial tremor sort of arising there. So it's a very rapid increase in movement problems in that last 2 years. For people with dementia with Lewy bodies, the movement doesn't change so much, but what really changes for them is their cognitive status. So people with Lewy body disease are early on starting to notice problems with their memory and so on, and that becomes more of their dominant problem. So this is really giving us a good insight into what is happening in Parkinson's before diagnosis. And it really is, this is giving us some hope that when treatments finally come along that might be able to slow down the disease process, we'll be able to identify people at risk and be potentially giving treatments up to sort of 10 years before people would traditionally get diagnosed. And so if and when there is going to be a treatment for Parkinson's, it's starting to be seen to be possible that we could actually stop the onset of the disease itself. So we don't have to wait for the motor symptoms to arrive, by which time a lot of the damage is already done. So potentially, as I say, if new treatments arise, we could be doing the impossible and treating the disease before it gets diagnosed. All right. So the other thing that Roger Barker talked about was that there are many subtypes of Parkinson's. So it's quite a complicated disease. And again, for the people in the room who have Parkinson's and for your partners, this won't be a surprise at all, because if you've talked to anyone else with Parkinson's, you'll know their experience is probably quite different. Everyone has got a different cluster of symptoms. Some things change more rapidly than others. And so it's a bit of a chameleon of a disease. And so, yep, it's not at all revelation for people here. There's a lot of variation across people. And can we make any sense of it? And this is sort of brand new research, really. It's just come out of the last year from a group in Europe that actually we do some collaborations with. And they have been looking at imaging of the brain, but also the whole body, and looking at changes in the gut and in the brain. And have come to the idea that we can potentially divide Parkinson's into two classes, what they call body-first Parkinson's and brain-first Parkinson's. And this undoes a little bit of what I was talking about before. But the idea here is that for people who've got body-first Parkinson's, the disease is potentially starting in the gut. And it passes up from the gut, from the vagus nerve, basically from the gut, up to the brain. And that follows that progression that we said before. It goes up through the spinal cord, up to the brainstem, and eventually gets to wider parts of the brain. The opposite may happen, however, for people who the disease may actually start in the brain and potentially in the nose, and then go down through the brain and backwards into the gut. And this is a relatively new conception here, and it still needs to be tested, I think, in other groups. But the difference between these two parties is that the people with body-first Parkinson's are the ones who have the REM sleep behavior disorder. And the people here who've got the brain-first Parkinson's are the ones who don't. And I think that makes a bit of sense here, because when we talk about REM sleep behavior disorder, certainly not everyone with Parkinson's will experience that. It might seem a bit of a strange thing to focus on. So there's a couple of things here. Why is this important? I think it explains a little bit about why, again, the disease is different in different people. But also it shows that we shouldn't put all our eggs in one basket. By focusing on that sleep behavior disorder, we may miss a bunch of people who go on to get this form of the disease. So why is it important? As I said, sleep behavior disorder isn't everything. We could miss a lot of cases there. But the other thing is that genes are important in a small number of cases. And increasingly in Christchurch here, it's going back to what you were talking about, blood types. We are taking blood from a lot of the people who come through our research program, so at least the people in Christchurch, getting a lot of genetic information. And we are finding some of our people with Parkinson's have specific genes that might be related to the cause of the disease in that person. So genes certainly aren't the be all and end all, but there's a variety of genes that seem to be closely related. And when we know that there's a particular gene involved in a particular case, that again allows us to understand how the disease is evolving, perhaps in that particular person, and means that there are different treatments that could be targeted to people with that particular gene. And I'll talk a little bit more about that as we go through. All right, so the other thing that Roger Barker talked about that's changed is advanced experimental drugs. So for people with Parkinson's, the primary gold standard of treatment is still levodopa, and it's been that way since the 1960s. You know, one would have thought that we would have, you know, there's been advances around the edges, but fundamentally replacing dopamine is still what we look to, and that does give good symptomatic relief, but it's not really treating the disease itself. So the idea here is that we now know that there's this alpha-synuclein prion floating around, and so there's this whole field of what's called small molecules and ways in which they can target that, and so there's a lot of research going on to do that. We've seen similar techniques happen just over the last year with the COVID-19 new vaccines, completely new ways of designing vaccines, and that's a similar sort of approach really, and so a lot of research is going on in that realm. It's happening in other diseases. People are already trying that in Huntington's and Alzheimer's. Unfortunately, none of those trials have really been successful, but they're continuing to evolve, and hopefully Parkinson's will learn from some of the results that have happened there. And ideally, when those treatments come out, as I say, we want to apply them before people have their symptoms. So that's one side, which is a brand new form of designing drugs. But the other side is, let's look at old-fashioned drugs and potentially use them for other purposes. That's something called drug repurposing. And that's something that Daniel's research potentially gives us a bit of a handle on too as well. Because there's a whole swag of drugs that people take just commonly as they get older, which have been shown to actually reduce the risk of Parkinson's. So we'll just go through some of those common ones. This actually is not so common, but it's something called ambroxol. And what that's used is for respiratory diseases. And it's really just to break up phlegm. So it's really a cough treatment, or for people who've got a lot of buildup of fluid in their lungs. But that seems to be particularly useful in around 8% of Parkinson's cases, which are related to something called the GBA gene. So this is the largest gene that seems to be responsible for Parkinson's. And we've been doing a bit of work here in Christchurch recently with taking DNA from most of the people who are coming through our research program, and have identified a small number of people who do have that GBA gene, which means it's likely to be responsible for Parkinson's in that particular person. And there's a hint there that this otherwise common drug may be useful in undoing the effect of that gene. So the possibility here, as I said, that we may be able to target particular treatments to individuals rather than trying it on everyone with Parkinson's. It almost certainly wouldn't work. But increasingly, we're getting to the stage where it might be possible to find specific treatments for specific people. Let's look at another one called exenatide. That's a treatment for diabetes type 2. Diabetes is increasingly common, and again, there does seem to be a bit of a link between diabetes and Parkinson's. And this is a drug that seems to work on what's called the gut-brain axis, which is the way that the gut and the brain talk to each other. And there are trials underway at the moment to see. There's been some promising results in animals, and again, we're still trying that out at the moment in people to see if that might have an effect. Another one, this is quite a common drug, is retipene, which people in the room may be taking. As we get older, it's quite a common calcium channel blocker, so it's used to reduce blood pressure. And it does seem, from looking at big population-wide studies, the sort of things that Daniel's involved in, or we're looking to be involved in, that this may again reduce the risk of Parkinson's disease. And there's been really good evidence from epidemiology, other studies around the world, showing that people taking isradipine as they get older are much less likely to get Parkinson's, or slightly less likely to get Parkinson's. Really good evidence from animals. So looking at mice, and good old friends, mice in labs, also seem to give really strong evidence that isradipine might be useful to prevent the changes involved in Parkinson's, but it has been tried in a few human studies so far, and the results haven't panned out. So it's always, it's a continual challenge in the Parkinson's drug development world. There's been lots of studies that work really well in animals and then fail when they're tested in people. There's still a lot of thought there that might be about the dosing not being correct, or maybe again it's being tried too late in the disease. So promising evidence there, but unfortunately nothing yet to show that that's going to be a useful treatment at this stage. And this is probably one of the more common ones. I suspect that a few people in the room will be taking this. This is simvastatin, which is used to reduce cholesterol, to keep our the blood vessels around our heart nice and clear. That also seems to be associated with reduced risk of Parkinson's disease. Again, good evidence from epidemiology, good evidence from animal studies, but again, it's been tried in people so far and the results have not been positive. So again, another source of frustration. But it does seem to be that this is potentially going to be a really a fruitful area of continued research, because I think, again, some of these studies will still be about trying to find the optimum dose, maybe about giving these treatments to selected patients, again, looking at the genetics, or ideally, perhaps, again, targeting people at the very early stages or even in the pre-diagnostic stages. So that's just a bit of a feeling for where research is at in the wider sense, how things have changed in the last 20 years. I think at least we're starting to now see some hope that levodopa is not going to be the only treatment in town, and that things might change in a reasonable sort of time frame. So I'm just going to finish up really with one last, a bit of a sales pitch really, that a new small element of some of the research that we're doing. I think there'll be, looks like there's people in the room that have definitely been involved in our research. And one of the things you would have seen, or that you would have been asked to do, is this task. I'm very glad that by the time I brought up that video that I'm not wearing the same shirt, just to show that I'm doing some changes. But you would have done this task, no doubt. This is part of what we call the UPDRS, or the Unified Parkinson's Disease Rating Scale. We ask people to open and close their hands, open and close their fingertips, do these sorts of things. Now, traditionally, in all the research that we've been doing in Christchurch, we keep all the data anonymous. But what we're going to ask people to do is certainly not compulsory, but we're asking people to be prepared to actually be videoed doing this task for a change. Because what we're looking at is, can we use artificial intelligence techniques to automatically diagnose and track motor behavior? So this is same video. This is me having passed the video to a clever artificial intelligence algorithm, and I'm just asking it to be able to track my fingertips and also my lips, because again, we sometimes see for people with Parkinson's that when you move your hands, there'll start to be associated movements of the mouth. And so you can see that it does pretty good. Actually, this is just relatively low quality video just from a cell phone, and this does pretty well. Looks like I've got really terrible nail polish on, but yeah. Anyway, just something for those of you who are involved in our research program, we will be asking the next time you come in if we can video you. You certainly don't have to say yes, but we would like to be able to get a body of video recordings like that, partly to see whether or not we can supplement the clinical examination. Normally what I would do is look at someone doing that task and just rate it from 0 to 4 on a particular scale. That's very subject to judgment. Someone else would give a different rating. But we're also, particularly after the COVID-19 kind of experience, looking at can we do remote monitoring or assessments of people, again, over their own phone, potentially. And this might also enable people to be involved in trials across the country. They don't necessarily have to be in Christchurch, and so we may be able to extend some of the involvement of our research wider afield into the country. And this also could be potentially used in drug trials so that we can do really easy monitoring of how effective medications are. And that's the kind of thing that we can easily get some numbers out of here, basically, just to track the limb and various movements. So I'm just going to wind up there. Happy to take any questions, but that's just sort of a quick overview of how things have changed in the Parkinson's world over the last few while. And I think that there is some hope out there for future change. Unfortunately, it is still relatively slow. I don't think we're going to see substantial new treatments or cures in the next 5 to 10 years, but I think that there's good evidence that there are a few pipelines that people are exploring. And I think there's reasonable hope over a longer time frame that we'll finally start to see some changes.